Hello everyone, my name is Valentin Kliebe. I'm a senior here in the biology department at Presbyterian College, and I performed my honors research with Dr. Waylay from the pharmacy school. I've worked with him for about a year now, and on the honors research project particularly, I worked with him the last semester and this semester. Um, we worked on the investigation of the effect of glutaminase inhibitors on chemotherapy-induced peripheral neuropathy. First off, I would like to start with why cancer-related pain is so significant and why is it so important to be concerned about it and do research about it. Cancer-related pain main causes include the underlying cancers itself as well as the side effects of treatments such as chemotherapy treatment and opioid treatment. For example, the treatment with morphine causes adverse side effects such as addictions and respiratory depression. If you look at the graph down here, we can see that over 50% of all cancer patients asked in this study reported pain related to their experience with cancer. And then surprisingly or very interestingly, every third patient reported pain after as being a cancer survivor after a completed successful treatment with cancer. And then about 60% reported pain for when they are undergoing treatment or they are in advanced metastatic stages or even in terminal stages of the cancer. In our study, we're particularly interested in neuropathic pain, which is one of the main side effects of many chemotherapy agents that are used to treat cancer. And neuropathic pain is caused by the damage to neurons located outside of the spinal cord and the brain. Neuropathy is a pain that usually occurs chronic, so it's a long-term issue for many patients. And as of right now, we really are lacking efficient approaches for the management of neuropathy. Since we're working with glutaminase inhibitors, I would like to introduce the enzyme glutaminase to you. And glutaminase is an enzyme that's located in the mitochondrion of the cells, and its main function is the catalysis of the conversion of glutamine to glutamate. In this diagram shows a tumor cell, and the glutaminase here is shown abbreviated as GLS, and glutamine is introduced into the cells where it gets converted by the enzyme glutaminase into glutamate. And glutamine is a precursor for many different metabolic pathways that are beneficial for cells. However, in elevated levels, and especially in tumor cells, it is very dangerous and it feeds the tumor cell with energy. Glutamate is a precursor for the GRAB, so TCA cycle, which it enters as alpha ketoglutarate and then eventually gets spit out as ATP, which is obviously very important for the excessive tumor growth. It also is an intermediate for many in protein synthesis and in biosynthesis, and acts as a precursor for many nutrients that are important for tumor cells in order to keep up their rapid growth. So those functions show why glutaminase is such a critical enzyme in the development of tumor growth. However, and then additionally, extensive levels of glutamate in the cells are leading to neuronal damage, and the excessive upregulation and expression of glutaminase is why it makes a critical enzyme for the development of tumor growth. However, as of right now, the impact on cancer-related pain remains still unclear. So we don't have much research on the effect of glutaminase, the direct effect of glutaminase on pain in cancer, which is why we in our study are looking at the nuclear factor erythroid 2 related factor 2, or short NIF2. It's an antioxidant signaling pathway that has shown and has been researched very well and has shown in many different studies that it is able to reduce cancer pain, particularly in neuropathic pain. It has shown to be able to do that, and It's also able to reduce chronic pain and chronic inflammation, on also neuroinflammation, which can be seen here in the diagram right here. It also has beneficial impacts on oxidative stress and mitochondrial dysfunction, which all are contributors to chronic pain. Under regular conditions, the NRF2, which is a transcription factor, is inhibited by the cage-like ECH-associated protein 1, or KEAP1, here in yellow, and is usually inhibited under function. Then when it gets activated, either by oxidative stress, inflammation, or by outside NRF2 inducers, such as here, NRF2 dissociates from KEAP1 and translocates down into the nucleus, where it binds to the promoter region of the antioxidant response elements, which then upregulate the expression of the AIE downstream proteins, which include the hemoxygenase 1 or the NADPH quinone dehydrogenase 1, which are both proteins that have an inhibitory impact on oxidative stress, neuroinflammation, and mitochondrial dysfunction as well. However, as of right now, there is no correlation or direct correlation been proved between glutaminase activity and NRF2 activation. So now our study comes into play. Actually, this data right here is from a study that we did last year, and we published actually last year in a paper towards the end of the semester, where we investigated the impact of the well-characterized glutaminase inhibitor compound 968 on NRF2 activation. So we were trying to make that connection of between glutaminase activity and NRF2 activation, or the inhibition of glutaminase activity and NRF2 activation. So in this graph here on the left, it shows the activation of the NRF2 signaling pathway. We see that our compound C968 was able to significantly increase the activation of NRF2 compared to our other groups that we use as control groups. They include sulforaphane, which is a very well-known and commonly used NRF2 inducer, as well as telaglenastat, which is a chemotherapy medication. It's used in chemotherapy and cancer therapy, and has anti-tumor activity. So we see here that C968 activated NRF2 activation, and then we looked at the downstream proteins, at the expression of the downstream protein, to investigate them as well and see if they actually get translated and expressed. So heme oxygenase 1 was significantly upregulated through the treatment with C968, as well as expected, honestly, in the treatment with sulforaphane, which is the NRF2 inducer. And we see it's a very similar pattern for the NKIR1 expression, where sulforaphane again dramatically increased the expression of NKIR1, but C968 as well significantly increased the expression, which is very promising, and which is actually the underlying basis for our studies in our honors research and for our animal model. So based on this data, now we want to take a step further and trying to determine the impact of the glutaminase inhibitor compound 968 and analogs in order to optimize its function on pain and opioid signaling pathways in cancer-related pain models. So what we did in our in vitro models is we seeded the cells in 96 well plates for 24 hours, and then on the next day we serum stuffed the cells for 1 hour, and then eventually treated the cells with the glutaminase inhibitors for 3 hours, and then performed our initial drug screenings by the use of the luciferase assay. Down here I've included the reporter cell line construct that we used, that was provided by Dr. Valentin from the University of Missouri. And so how that functions is NF2 gets activated, dissociates from KEAP1, and then translocates into a nucleus where it binds to the promoter region. Those cell line constructs are stably expressing the antioxidants response elements. So it binds to the promoter regions, and then by the binding, it promotes the expression of the luciferase gene, and eventually the expression of the luciferase enzyme. And then the luciferase enzyme has luminescence, which we measure on our mini plate reader, and that gives us an indirect measurement on NRF2 activation. Our experimental designs in the in vivo models, so we use two different main experiments in our in vivo models. The first one where we treated the mice simultaneously with the paclitaxel, which is a chemotherapy agent that is used in cancer therapy in order to induce the pain, and then we treated them simultaneously with the glutaminase inhibitors. So paclitaxel is commonly used in chemotherapy, and one of its main side effects is the neuropathic pain. And on day one, we performed the one-for assay to establish a baseline of the mechanical threshold. And then right after, we injected the paclitaxel and the glutaminase inhibitors into the mice. And we did the same thing again on day 3, 5, and 7, shown here in blue, to develop our pain model. And then on days 4, 5, 7, and the final one on 8, we performed the one-for assay again to measure the mechanical threshold and measure the sensitivity of the mice to the pain. And in our second experiment, we again started with a baseline on day 1 without any prior treatment, just the mice without anything. And then on right after, on day 1, and again on day 3, 5, and 7, we injected the paclitaxel. And this time we did not simultaneously treat them with the glutaminase inhibitors. So we induced the pain for 7 straight days by the injections on day 1, 3, 5, and 7 to develop the pain over this week. And then after this week, on day 8, we performed the one-for assay to get a second baseline to confirm the pain has developed. So in that baseline, the mice are supposed to show a way higher sensitivity to the pain. And then right after that, on day 8, we treated the mice with our glutaminase inhibitors for about 24 hours. And on day 9, we injected morphine into the mice at a concentration of 3.2 milligram per kilogram body weight, and performed the one-for assay multiple times over a 3-hour time course to investigate the impact of our glutaminase inhibitors on the analgesic effects of morphine. So what is a one-for assay? In the one-for assay, we used it to measure the mechanical thresholds where the mice exposed to. This picture down here is actually taken in our lab. It shows 3 different treatment groups with 5 mice in each group. One will be the vehicle, one will be the compound C968, and then one other compound that we chose based on our in vitro experiments. So the mice are placed in individual boxes on this mesh-like surface, and then those are the filaments from our lab, and those filaments have different strengths. So using a higher strength of filament means higher mechanical threshold, so the mice experiences less pain if it can go up to those stronger filaments. So we use the filaments to poke the mice into the left back paw and see if they respond or not, and based on if they respond or not, we either go down or up to poke them again with a different strength. And based on that, we establish our mechanical thresholds. In our in vitro models, we investigated the impact of C968 analogs on NRF2 activation. So in this graph, we can see that C968 confirmed our results from our study that we published last semester, where C968 increased the NRF2 activation, and did it again here, just as expected. And then on this slide, we see a whole bunch of other compounds, analogs of C968. And out of all these, we see that the XC0115, which is marked here with the red arrows, was able to increase NF2 activation. And very interestingly, it pretty much showed the same effect, or almost the same effect, on both concentrations. On the high 10 micromolar concentrations, where there were also a couple others that showed an interesting activation on NF2. However, none of them also showed a high activation on the 1 micromolar concentration, which is why we selected, along with C968, also the XC0115 compound for the animal models. And from there, we moved into our animal models, where we see that here in the left graph, in the male mice, we see that the treatment with both glutaminase inhibitors, the dark spots, C968 in the circles and then XC0115 in the squares, was able to actually significantly increase decrease the sensitivity to pain in our mice on day 8, and also on the days before it was able to decrease the sensitivity of our mice to pain by the treatment of glutaminase inhibitors. So we can see the vehicle has a lower mechanical threshold, which means filaments that have less strength already caused pain or responses by the mice, compared to our treatment groups, where we needed filaments with more strength in order to get a response, which means they were less sensitive to pain. Unfortunately, we did not see the same pattern in our female mice. We don't know exactly why that is, but we'll keep doing those experiments and repeating the experiments and investigating if that pattern is confirmed, that there are differences between male and female mice, and investigate why that could be. This is our second main animal model, where we see that our glutaminase inhibitors were able to enhance the morphine analgesic effects in chemotherapy-induced peripheral neuropathy. We see that this is the first baseline without any treatment. No morphine, no glutaminase inhibitors, nothing. And then after the 7 days of paclitaxel injections, we did another baseline, which is obviously very, very lower. So the mice are very sensitive to pain right now because of the paclitaxel injections they have gotten throughout the week, induced the pain. So we confirmed that our pain model works, and then we injected morphine and performed the 1,4-ASM multiple times over this 3-hour time course. And then we see that morphine alone, here in the black squares, did not show a huge effect on analgesic effect on our mice, which is not very surprising because morphine does not work very well in neuropathic pain, because it works through a little different mechanism, which does not really apply to the neuronal damage that is caused to neuropathic pain. But we see that the co-treatment with C968 here in blue, and for XC0115 even stronger, significantly increased the analgesic effects of morphine, which is very promising, because if you think about it, if you need less morphine in your treatment groups, that would also mean a reduction in side effects of morphine, which can be very adverse, not just addiction, but also, or mainly, respiratory depression, which often limits doctors in the treatment with morphine because of the risk of that respiratory depression. So these were very exciting and promising results that we got here. from there, we have one more pain model, which is the TAFLIC assay that we performed. It is an acute pain model, so a little different than the pain models we've used before, where we hold the mice tail in a 52 degrees Celsius warm water bath and wait for a response, or wait until it spontaneously flicks out the tail because of the uncomfortability or pain it feels. The cutoff time is 10 seconds. We don't go over 10 seconds because there could cause tissue damage in the tail. So perform the baseline and then treated the cells for 24, the mice for 24 hours with our glutaminase inhibitors of C968, and then injected morphine and performed the TAFLIC assay again over a 2-hour time course. And we also see that the compound C968 was not just able to increase the analgesic effects in neuropathic pain, but also in this acute pain model. So our inhibitors work in different pain models, which is very exciting as well. In summary, we can say that our compound 968 and XC0115 increased NRF2 activation in our in vitro models, and then based on those screenings, we took the C968 and XC0115 into our animal models, and we saw that the treatment with both of them enhanced the morphine analgesic effects in chemotherapy-induced neuropathic pain models. So in summary, we can say that glutaminase inhibitors potentially able to slow down the development of chemotherapy-induced peripheral neuropathy, which is a very pleasing result of our study, and we'll keep doing the experiments. We are not done yet. We have to repeat experiments and confirm our results, which we will be doing in the next weeks. And last but not least, I want to say thank you to Dr. Lay, my faculty advisor, my mentor, doing this research, for sharing his expertise with me and his exceptional mentorship. And also want to say thank you to Deltrice Holmes, senior here in the biochemistry department at PC, who worked with me in the lab this and last semester, and then to our collaborators, Dr. Chen from Campbell University, who created and synthesized the inhibitors and the analogs of the C968 for us. And then Dr. Stryker from the University of Arizona for contributing data, and Dr. Valmosin from the University of Missouri for providing the ARE report to cell lines. Thank you very much.